I had the pleasure of introducing Dr. Anthony Leonardi, who on December 21st, 2020, published a groundbreaking study regarding the adverse immunological impacts of COVID-19. The paper, along with subsequent research, described aberrant T-cell differentiation and apoptosis, which is programmed cellular death, in COVID-19 patients. Dr. Leonardi's presentation will provide a cursory look at COVID-mediated immune dysfunction. This presentation will explore the adaptive immune impact from SARS-CoV-2, the early clinical signals of immune harm, underlying mechanisms, real-world presentations from viral reactivations to decrease pathogen surveillance and control, and what this may mean in terms of prognosis and future interventions. Dr. Leonardi, the floor is yours.
Thank you so much, Dr. Kavanaugh. Thanks for the invitation to present to everyone here today. Yeah, let's get right into it. And about that publication that you mentioned, it was a highly controversial claim to say that the lymphopenia was apoptosis, was the cell death. But from what I understood, I was pretty certain it was, and that's why I postulated it. And that was subsequently found by a Canadian group that it was indeed apoptosis years later. So the lymphopenia does indeed come with apoptosis. So just to start off with disclosures, I've received funding from Canthro Philanthropic Fund, and that is for T-cell work. That philanthropic fund is a fund started by Vitalik Buterin. He allocated money and has given funds to UC San Diego for clean air, PolyBio Foundation, which is investigating long-term SARS-CoV-2 persistence and other immune effects. And they also reached out to me. So yes, I'm working on that. I funded, to operate with that grant, I funded CoreImmune, and that work is open source from the Canro Fund. So it's open source work right now. So my background, I was at the National Cancer Institute from 2010 to 2013 as a post-baccalaureate fellow, kind of floating around projects, helping postdocs. I had my only idea, I had the bright idea to look at T-cell death and differentiation as possibly a linked mechanism, and I did find that. So we'll go over that, and then we'll go over the early evidence of T-cell death from the Wuhan cohort that was published in The Lancet under, I think it was Richard Horton as the editor. We'll talk about the mechanisms of the immune damage that occurs from SARS-CoV-2. We'll talk about the presentation that someone may see from subsequent damage from SARS-CoV-2, what you might see if you're encountering this population of people, which is pretty much ubiquitous. Many, many people are out there. And we'll talk about what potentially it has as far as an epidemiological impact on other infections and what it could mean for also elderly populations, also some information about what's going on in children and what a prognosis could be in the future directions, kind of a call to action and focus. So part one, the background. So my research background was on T-cell death and differentiation. And what that is basically how a T-cell goes from being unstimulated, naive, not ever having encountered antigen, to getting stimulated, traveling to where it needs to go, and dealing with antigen or reacting in some way. So basically, what I found was that there's a canonical death receptor called CD95 or FAST receptor. And when you agonize that receptor, it's not just cell death. In fact, it's T-cell differentiation, which means that T-cell changes, matures, and then becomes lytic and becomes cytotoxic. Okay, so basically what was found was that in CD8 T cells, if you stimulate the CD8 T cell and agonize FAST receptor, you basically just get further differentiation, and then all the apoptosis is in the effector cells. So it's like a life cycle, and it's very elegant that it's linked in one molecule. It goes from, naive central memory, effector memory, then you get the apoptosis. Now, going over to how this was kind of found was I kind of had a pet interest in Ebola virus. The virus is never found inside the T cells themselves, yet what happens in infections, and especially severe infections where the patient dies, is there's a massive lymphocyte death and, of course, the hemorrhagic syndrome. And death is correlated with the amount of T cell loss and lymphopenia and other protein markers. One of them, namely, was Fas ligand, soluble Fas ligand, and Fas receptor, soluble Fas. And so there are a few papers. There was this really excellent paper that I read, Does the Virus Toll the Bell for the Immune System? And they pointed at Fas and Fas ligand, as some sort of marker of severe Ebola or fatal Ebola. And what we had observed in the laboratory was that there was this phenomenon where, and this was observed by a Dr. Christopher Hinrichs, who then gave the project to a Dr. Christopher Scott, that basically when antigen experienced cells, like central memory cells, are stimulated contemporaneously and in the same space with naive T cells, those naive T cells then differentiate much faster. Okay, they grow up and start killing much faster. And so they were focusing on what is the mechanism. And I thought, well, okay, so they're growing up faster than dying. Maybe it's this Fas ligand. So Ebola basically just massively activates CD4 and CD8 T cells, upregulates Fas and Fas ligand, widespread T cell death despite the T cells never being infected. And where I come in was that T cells differentiate via Fas before they undergo programmed cell death. Part of that paper that we published, it didn't include Ebola virus as part of the rationale for investigating Fas. We just said, hey, we saw Fas was differentially expressed on microarray. It was a much simpler explanation than going through like all this other rationale. So there's this quote, this was once revealed to me in a dream. This was revealed by a microarray. So it's interesting, part of the narrative is lost. So now going to the pandemic of the signals of T cell harm. So in the beginning, that paper in the Lancet by Huang et al., there was profound lymphopenia in a lot of patients. And to me, that means, yeah, sure, massive activation and movement to the periphery, but also with that, because I know the life cycle of a T cell, I know that the T cells that hang off in the effector end are going to go through apoptosis and cell death. So wait, hold on, it's not just broad stimulation. It's very likely there's going to be a cell death component. So a few more papers came out with lymphopenia as a strong correlate of disease severity. There was definitely a correlate of either stimulation or the immune system kind of giving up the ghost, as it were. So in May, there was a preprint. It was eventually published in 2021, but in May 2020, there was another preprint showing there was a very broad stimulation of the T cells in patients and also a significant reduction in the proportion of naive cells and increases in effector T cells. When that happens, when that occurs, those T cells are going to be lytic, they're going to be causing harm, cytotoxic T cells going to kill other cells. And it's just going to create like this massive inflammation and cell death and also immune aging because, remember, you start off as a naive T cell, then you go to central memory and effector memory. If you kind of move that needle and push the T cells along in their lifespan, it's going to change their dynamics. And a loss of naive T cells, like it's not just the SARS-CoV-2 specific T cells, it was broad, which was very interesting and which was a topic of great contention at first. The question is, how is SARS-CoV-2 massively stimulating the T cells? It's a virus. I mean, so does Ebola. Ebola has a canonical superantigen, which means it can broadly stimulate T cells. So what's going on with SARS-CoV-2? Nevertheless, despite all the mechanistic arguments of how it's happening, it indeed happens because we can observe it. Just going on to not only cell death, but T cell exhaustion, groups also found that there was high PD-1 expression. So this is canonically the exhausted T cell, the T cell that is done being stimulated. It does not want to react. And this was also paired with high CD95. So to me, it looked like this infection was very, very hard on the immune system, hard on the adaptive arm of the immune system. And I think, in some sort of effect, like that was kind of inconvenient information, right? It wasn't too hard to publish initially, but it was difficult for people to say this is indeed significant and could be clinically significant in the future. So Motorbacher, part of Shane Crade's group at UC San Diego, they did an analysis and found that naive T cells are protective during acute COVID. And so you can see an association. The more naive T cells you have, the better you'll fare. And there could be a number of reasons that is. Naive T cells, they help you respond to infections, but they might also do some other regulatory thing. And that's what I seek to investigate with Conroe. But indeed, it's a correlate, and there are different rationales for that. In mid 2020, I was saying, like, the stimulation is so broad and it's so heavy. I really hope that it doesn't eat up our naive T cells. I made several public statements about this. I hope it doesn't just broadly activate, cause cell turnover, because we don't need turnover in the adaptive immune system. So it's not only acute COVID, as Motorbacher showed, but Petsupan over in Australia. And what they found was that, and it was published in 2022, that patients with long COVID are missing the subset of unactivated naive T cells and unactivated B cells. It was actually quite shocking. They did follow up with the patients again, and that subset reconstituted after a few years, which is good news. But I'd still be wary about how good news that is given the constant reinfections that we're encountering with SARS-CoV-2. But that was reassuring. But nevertheless, there was turnover happening. Either turnover, well, it had to be turnover because it's a paucity of unactivated naive cells. So the only way you're going to lose those is if they're being activated and then not replenished enough or at all. So not only does long COVID, represented by this loss of T cells, and not only does SARS-CoV-2 cause a lot of lymphocyte differentiation and apoptosis, but also it does something called hyperactivates T regulatory cells. And these are cells responsible for quelling autoimmunity. And it's a therapeutic target for cancers. If you can block the tolerogenic cells, then you can kind of unleash the autoreactive anti-tumor T cells. So that is a target, but what SARS-CoV-2 will do, especially in severe cases, is it turns your T regulatory cells into what they coined hyperactivated T cells. And these will license autoimmunity instead. And so in 2021, after reading this paper and incorporating it into, well, actually 2020, Frontiers article, I said there's going to be a lot of autoimmunity coming because this change is going to happen. They asked what sort of diagnoses. I said, I don't know. Global, I think. I think it's going to be global because this is how we fight autoimmunity and it's being distorted. And unfortunately, there are massive increases in autoimmunity now. Also, there are antigenic reservoirs of this virus. And what was found was, just a study on adenoid and tonsils tissues from kids, they found even in kids with no documented prior infection, they were finding SARS-CoV-2 proteins. The nucleocapsid is not present in the vaccine. So it is not the vaccine. And some of the proteins were inside T cells. Even if it wasn't infecting the cells proper, there's still a broad stimulation happening. So I'm not sure if most of the T cell stimulation or apoptosis is because of infection of the cells. That's still contentious despite being published by Shi Zhengli in a Nature Family Journal. That's still contentious whether or not the T cells are being infected. Nevertheless, we can find antigenic reservoirs in previously quote unquote uninfected children. Maybe they were exposed, maybe it was subclinical, hard to say. And then Risa Pretorius's group, along with Rob West, they wanted to study long COVID and ME/CFS versus healthy donors and see if there was a difference in the nucleocapsid protein in the muscles of these people. But pretty much even the controls that didn't have long COVID had nucleocapsid protein in their muscles. So it's ubiquitous and it poses an interesting challenge, the fact that it's so ubiquitous that there are echoes of infection within so many populations. It makes it hard to control. But I'm going to come to a study by Peluso et al. That gets around this in an interesting way. And there was another piece of evidence about the antigenic persistence. It's remaining in the gut, and it can be found in the gut up to the time point of when we look now. It can be found from someone that has a documented infection a long time ago. The problem is we don't know if someone was reinfected, right? But nevertheless, it's persisting in the gut, and it's persisting despite where you would expect there to be some turnover. And of course, the nucleocapsid is not present in the vaccine. So it is not the vaccine that you're seeing in the gut. These images were of SARS-CoV-2 virus specifically remaining in the gut. So going back to that study by Peluso et al. At UCSF, they found pretty much everybody currently has T cells that seem activated according to like a type of PET scan that they're using that can measure T cell activation. Pretty much across the board, we're seeing T cell activation. So it was really hard for them to compare between healthy donors and individuals with long COVID. So what they did was they looked at historical scans, and they found out that there was a significant difference in the current population today broadly versus their historic populations before SARS-CoV-2 infections occurred. So basically, there was broad T cell activation in the world today of their population, regardless of whether or not they had long COVID, versus historical controls before COVID was ever found. So this broad T cell activation, possibly turnover, apoptosis that's occurring, not in terms of a massive acute infection that was evident in those early publications in 2020, but rather now where there's this smoldering activation, it presents a different situation. And that's why I want to bring this into the post-COVID immune dysfunction. A lot of people will focus on the term post-COVID because they feel like it could be potentially chronic infection, or you never are post-COVID. But for now, it could also say COVID-mediated immune dysfunction. That's up to everybody else. So just to kind of provide a framework for myself to understand it, basically, you're getting a premature aging of the immune system, of the adaptive immune arm. The T cells are differentiating very broadly. That's evident. There was a Frontiers paper that showed age-related T cell changes, but it's basically pushing the needle on the lifespan of the entire repertoire, I'd say. Because this is blood, they didn't select for specific T cells. It was just very broad shift, differentiation states of the whole. And indeed, I mean, it's published, maybe contentious. There's increased susceptibility to differing infections. The BMJ had something that showed invasive group A strep. There's also a higher risk of shingles after SARS-CoV-2. There is going to be impaired immunosurveillance, and there are a lot of NK people, and I wish I included more natural killer cell stuff, but indeed, there's NK cell dysfunction. And of course, this is important because the NK cells are going around and killing cells that aren't expressing MHC class 1. And one of the things that loses MHC class 1 is cancers. Another thing that loses MHC class 1 is a cell that's infected by SARS-CoV-2 because it suppresses MHC class 1 via ORF8 and other ORFs. And of course, there's awoken autoimmunity as part of the post-COVID immunodysfunction sphere. You have these changes to the T regulatory cells. We have this huge proliferation of autoimmune conditions that's evident now, unfortunately. So mechanistically, you have the individual, and that individual will have a SARS-CoV-2 infection. They can get lymphopenia. They can have some naive T cell loss and the broad differentiation that comes with it, like some T cell exhaustion and immunoparalysis. They can also have antigenic reservoirs there, and they'll just have chronic immune dysfunction. There were some really nice papers that I didn't have time to include, but one of them shows T cell exhaustion subsequent to SARS-CoV-2 infection in moderate cases, and that exhaustion translated to infections of other types, other antigens other than SARS-CoV-2. And in the population of SARS-CoV-2 mediated immune dysfunction, in the population level, and this is the thing that I kind of like to think about, You have a population with widespread changes to their immune system, maybe a little bit of T cell exhaustion, maybe a little bit of paralysis, and you have the R value of other pathogens. And if there is a hit to collective populational immunity and disease control, you might change the R value. All things being the same, the R value might change because of impaired, let's say abortive cases. Let's say someone who was previously going to be an abortive case of measles or something. Now they've got a small hit to their immunity, so now it's not fulminant, but it's there, it's contagious. So you got immune attenuation subsequent to SARS-CoV-2, poor pathogen control, a reproduction number shift, and then potentially an outbreak. David Joffe, I have to mention him because, controversial, and he thought it'd be funny to name it the Leonardi effect. And then other people said, oh, no, the Leonardi effect is something else, trying to, make a joke, which is funny. But I just got to name him because he's recognized this as a potential effect as well. So there's an opposing theory to this, and it was platformed everywhere, especially the BMJ, called immunity debt. And the concept was this. Basically, because everyone was kind of on hiatus from getting infected with just normal pathogens, their immunity kind of waned because they didn't have the chronic stimulation and then immune top-ups, et cetera, immune reactions that would then boost titers. So the rationale was that surging diseases after SARS-CoV-2 in the population were indeed not representative at all of immune harm, but were rather because everybody synchronously got infected because everybody's immunity waned, and then there was overshoot. Now, this might explain in the acute setting, like 1 or 2 years, this might explain surges of these pathogens. But the problem is, when we look at what could be going on, there's a wealth of information about immune system changes that are occurring, and we seem to never pay off this immunity debt. Infections, they keep spreading. We see viral reactivation of certain viruses in people. We see things like other conditions increasing, other reactivations increasing, and we know, like on the individual level, that EBV reads, Epstein-Barr reads, go up during SARS-CoV-2 infection. And some people even have Epstein-Barr reactivation, especially if they're in a severe case. So this explanation may not hold for very long. We'll see. It may not be sufficient any longer. So going to a recent paper, basically, and what the striking thing to me from this paper is that this is only after one infection. And this is old information, but it's getting a lot of attention right now. Basically, after one infection, there's a reduction in T cell counts broadly, and it is very slow to reconstitute, if at all. And it's interesting because what I would like to know is after this third, second, fourth, Michael Horger over in Louisiana, he calculates some averages and what he expects the number of infections to go, people to have had by now. And I think he's saying like maybe around 2 or 3. I'm not sure, but yeah, so this is changing. But there's something very stark about this study that is important to share. There are conditions of chronic inflammation and metabolic syndrome, as we know, like patients with heart disease. It's well known in the literature of chronic cytokine expression and immunological aging and cardiac disease. These things are all part of a system. What this group found was that in cardiac patients, that hit to the adaptive system is hard and it's sustained. And their counts hit the floor and kind of stayed there. And that goes into the whole idea of T cells going through this lifespan of, they have naivety, then central memory, effector memory, apoptosis. When you age the T cells, they're going to be more effectory. You're going to have more effector-like population and more inflammation caused by those cells. And to what extent is that inflammation, that tendency of inflammation, contributing to cardiac disease? Or is it vice versa? Is cardiac disease, does it have a state of chronic inflammation that just like the T cells are just ready to collapse? Or should we just view it under the greater metabolic syndrome umbrella? But indeed, like somebody with these kinds of levels and somebody with these kinds of immunological changes, their immune system, I wouldn't be surprised if there was a predominance of shingles reactivation in these people. And this is the study that I was mentioning before. These are changes that are associated with age. So basically, after SARS-CoV-2 infection, people's immune systems, the CD4, CD8s, they looked older. They looked like they were from an older individual. And especially for the older individuals themselves, that effect was all the more dramatic. It pushes the needle along in T cell differentiation. Now, SARS-CoV-2 infection pushing differentiation so hard is different from just like infecting cells willy-nilly and causing them to apoptose. This is more pushing them to differentiate. And at the end of that differentiation pathway is indeed apoptosis. But there's going to be inflammation along the way, especially if there's a broad activation. So what all those mechanistic things, all those T cell characterizations amount to is, where do we see the pudding? Where's the proof? There's a group at, I think it was UT Austin, U Texas. Yeah. They looked at viral reactivation in patients with SARS-CoV-2 and they created a kinetic. What was interesting is they found there was one correlate with their patients they categorized as having long COVID. But indeed, there was reactivation of viruses that are normally controlled by the immune system. But this does indeed happen with other infections like influenza. But the strong T cell hit is not there in influenza. It's not like SARS-CoV-2 is much more potent as an activator of T cells. Was this the same paper? Yeah. So they tracked the specific viruses according to what type of long COVID that the individuals had. And for people with like a physical long COVID, there was a strong predominance of this virus called Anelloviridae. I think it was a gut virus. Oh, it was present in the PBMC. But this is a preprint. It's not published yet. But it's just another cursory look of where things are headed as far as understanding the complex interplay between SARS-CoV-2 and viruses that are reactivating and poor immune control. Here's a study that you might be interested in. Basically, they did a meta-analysis comparing herpes zoster recurrence after vaccination versus after infection. And of course, infection was associated with reactivation potentially, and vaccination was not in the meta-analysis. So the vaccination, it is a transient presence of mRNA or protein, if it's Novavax, that is then stimulating the immune system in proper sites. And then that stimulation then subsides. Whereas when you see the infection and you see the antigenic persistence and nucleocapsid itself persisting in patients, that is able to remain in the body and chronically activate. So it is indeed different. And that's why I would tell people, like, you want an immune response, you want it controlled and temporary, and in a place you want an immune response, like in your arm muscle deltoid. Where you don't want an immune response is in the brain, and that's where SARS-CoV-2 has been found in the brain. So we don't get vaccinated in our brains, fortunately. So indeed, another group found that there was increased risk of not only, well, it was medically attended RSV, so kind of severity, but if a child had SARS-CoV-2 prior, they were more at risk relative to a child that did not have SARS-CoV-2 infection. So there's some small signals there about potential decreased population surveillance or control, or what would have been an abortive infection, or maybe a strong reaction to infection because of sensitivity. This isn't elucidated by this study, but at least it's a signal. And again, going back to that BMJ study about group A strep, if you look on the right, there's the risk of specific conditions and infections and chronic conditions. And the strongest signals that were statistically significant, I wish I included the p-value, but at least there are the confidence intervals there, was in streptococcal tonsillitis, But yeah, they found group A strep was increased after SARS-CoV-2. So just going back to a framework to help me understand what's going on. So you have SARS-CoV-2 kind of pushing T cells down the line, kind of down a cliff, also causing T cell exhaustion. The aged population as a whole, if you aren't replenishing your T cells, if your thymus has involuted and you have the T cells that were kind of formed already, this could be more significant, and this is likely more significant in this population. And if reinfection and infections themselves occurs, this is kind of broadly differentiating and pushing those T cells along their pathway. And clinical consequences, you could potentially see more severe influenza. You do see more clinically intended RSV. You could see bacterial infections like the group A strep, and there were some other signals that we couldn't get to. But indeed, there's definitely a signal there versus the hypothesis of immunity debt. Another issue about possible decreased surveillance, the authors don't make any strong claims as to what is going on. They're only disclosing an observation that they made. But basically, in a group that chose to look histologically at cases of colorectal cancer in their IBD patients, they found SARS-CoV-2 antigen. And it was the spike antigen I looked. So they didn't necessarily look at nucleocapsid only, but they found in every single one of their cancers that they looked at, they found persistent antigen. And we can't say that it would be the cause or it would be pro-oncogenic or anything, but it kind of speaks to the persistence and that they don't really have controls as well. It's just an observation. But they do say one of the theories could be that it could be oncogenic in some means. If you were to ask my opinion, it would be that there's just the NK cells and surveilling T cells are taking a hit, and it is possible there's decreased immune surveillance. That's very controversial to say, but I would act with caution and consider that as a possibility. We discussed these three examples of increased risk of infection and increased severity. So, what can we expect? What's going to be the prognosis of someone with this? Well, let's characterize at least their T cell counts. Let's find out where they are. Let's find out if it's just a typical shingles case, or if they were, like, let's say, a cardiac patient and their T cells are through the floor. Let's at least know that and even be proactive and get people's white cell counts and figure this out. So for young people in mild cases and younger people, yeah, they'll have initial turnover and they might have a loss of inactivated naive T cells and B cells, but those did reconstitute in a later publication. But yet with repeated insults that we're having now and repeated infections of SARS-CoV-2, I wonder if you can't go back to that and push those T cells along the line. In the elderly, I think it's going to be more of a challenge because they don't replenish the T cells as readily. They have thymic involution, and they're also going to have probably a tendency for more fibrosis and just more severe cases. So I would be concerned. It's going to be like exacerbations of COPD. You're going to have to worry that they can potentially get infected again. Sure, they'll have the lung sequelae and lung issues, but then there's this immunological aspect to contend with as well. It's not really known how long susceptibility would last and how long these T cell changes are going to exist for. Going back to that study in the cardiac patients and the lymphopenia, there didn't appear to be a trajectory of replenishment. But of course, these things are nonlinear, so it could occur. It just needs to be followed up. Or it might be the case that something needs to be done in terms of antivirals or something else. There is a cumulative insult indeed from reinfections, and Dr. Al Ali over at WashU in St. Louis has characterized that very well. And it could be harm to the adaptive system for the repeat infections. So indeed, immune systems are compensating. We're making antibodies, we're addressing the new variants when we're rechallenged. But when one slips behind the defenses that we have, there's the potential for real immunological insult. So it's not really a known thing as far as clinical practice goes. However, I know in talks with many people, And there are many specialists, oncologists, infectious disease experts. They know sometimes lymphocytes are hitting the floor. There are consequences to that, but it hasn't been made into a formalized thing yet. And we currently haven't kind of adapted to the new normal of these immunological insults. So maybe that would include some immune panels in primary care. And this set of unknowns, we should probably take action and probably try to come up with solutions and anticipate how to surveil and how to figure out how to address this. So for me, or actually not for me, for everyone, so I'd love to see immune recovery, especially in those patients with metabolic syndromes and inflammation and aged patients. But we need to know what's happening. And it is being done, of course. We need the biomarkers to know who's going to have long COVID, or is there a biomarker for potential viral reactivation? We need to find therapies to replenish these stores. I know there is the owner of the LA Times, the doctor, surgeon, Patrick Soon-Shiong, he's talking about his company a lot and his therapeutic. We need more of that. We need more options. Me, myself, Volvi is also Counterro. They changed their name. Basically, I'm going to be looking at whether the loss of naive T cells represents a loss of a type of internal immune regulation or a systemic immune regulation of the T cells in the system. So I'm just going to be using murine T cells. This is going to be mouse work. And there are indeed a lot of suggestions from a lot of different parties about how this can be dealt with. I know some people were working on trying to make new thymi or thymuses, addressing T cell exhaustion with PD-1 or PD-L1 antibodies, vaccinating, kind of preempting the insults, IL-15, as we said, from Patrick Soon-Shiong, Dr. Soon-Shiong, and getting some new data and new information about how to address this in other models. And so, very pessimistic talk, I know, but COVID-19, SARS-CoV-2, it'll hit the adaptive immune system. Certainly used to be contentious, used to be difficult to say. Now it's obvious. There is naive T cell and activated naive T cell loss. There is turnover happening, broad turnover. The elderly, by virtue of the lack of replenishment and preponderance of more differentiated T cells, are going to have more evidence of immunodysfunction. But that's not to say it won't be more appreciated in the young, because in a lot of situations, the young are in situations where they might get more exposure. And we have the opportunity to actually make an effort and try to understand and potentially address this. So clinically, we can start looking at white cell counts and subsets. We can try to see if there's any evidence of reactivations in the elderly patients. People's EBV reads can go up, or their antibodies EBV can go up just inexplicably. But that always happened, but it may be occurring more. And of course, we can continue to support researchers and investigators and clinicians. And thank you all very much. I hope it wasn't too long or too long-winded, and I'll be taking questions.